In cirrhosis, systemic inflammation often progresses silently, only becoming apparent when patients reach a critical stage. We wanted to understand how systemic inflammation drives disease progression in acutely decompensated cirrhosis, and particularly how it contributes to the development of acute-on-chronic liver failure, or ACLF, a syndrome characterized by multi-organ system failure and high short-term mortality. In our study, we found that patients who develop ACLF had consistently higher levels of inflammation measured over time. This wave-like inflammatory trajectory could be key for identifying critical windows for intervention. However, standard biomarkers such as cytokines, white blood cell count, neutrophil to lymphocyte ratio, and C-reactive protein fell short to reliably identify patients who already had or were starting to develop ACLF at the time of hospital admission.
To go beyond standard biomarkers, we turned to drug findings. We analyzed blood samples from over 700 patients and identified 28 genes. That helped us better capture the complexity of the inflammatory states. That helped these genes form the basis of the CLIF-SIG score, which stands for Chronic Liver Failure Systemic Inflammation Gene Score. We then assessed how much each gene contributed to the score and traced their cellular origins. Thus, we developed a score that measures the intensity of inflammation and affects disease severity.
To bring the score closer to the clinic, we measured the expression of the 28 genes composing the SIG score using two different technologies commonly in place in tertiary hospitals. The first technology was NanoString, which uses the encounter analysis system and measures gene expression, the MRL levels, directly without amplification. The second one was FluidiGym, a high-throughput real-time PCR microfluidics technology that allows the analysis of gene expression and nanoliter scale volumes. In general, we observe a very good correlation of the RNA-seq data with the levels of the respective genes assessed in NanoString and FluidiGym platforms. Therefore, the SIG score has clinical applicability for its effective implementation in the clinic or hospital setting.
When we compare the CLIF-SIG score to standard clinical biomarkers using receiver operating characteristics analysis, a method that evaluates the test accuracy, the CLIF-SIG score demonstrated superior performance with a higher area under the curve, indicating a better discrimination between patients with severe and mild disease. Importantly, the CLIF-SIG score is not only accurate but also robust. It works independently of the cause of liver disease, the preceding event, even in patients with infection or alcohol-related hepatitis. In fact, 80% of the patients who developed ACLF during hospitalization had a CLIF-SIG score above the 0.386 at admission. We also validated these findings in patients with infections and sepsis and septic shock, showing this score isn't limited by diagnosis of liver disease. By tracking inflammation over time, the CLIF-SIG score could help guide treatment decisions early, before complications set in. It could also support patient stratification in clinical trial and improve how we assess response to anti-inflammatory therapies. This marks an important step towards precision medicine, with ongoing efforts to further develop and apply the CLIF-SIG score in clinical practice. Indeed, this score was cross-validated in a HPV ACLF cohort in China, as shown in a recent letter to the NHL.